I'm Brandon. I'm a postdoc with Hernan Garcia at UC Berkeley. And today I'm going to tell you about my work using fly larvae as a model to study immune responses and infectious diseases. Immune responses are inherently, by definition, dynamic phenomenon. They involve the coordination of thousands of different cells. So I think there's a lot of rich physics to learn here. I think physics has a lot to offer. And today I want to talk about just kind of initial ideas I have about how we go about building quantitative frameworks for connecting the behaviors of individual cells up to the macroscopic immune responses that ultimately determine infection outcomes. So I want to start with a historical example of where really simple coarse-grained mathematical models were incredibly useful in the context of infectious disease, and that's the case of HIV. All right, so many of you are probably aware of this work, but classic work, physicist Alan Perelson and colleagues developed a really simple ODE model for the total amount of HIV virus in a person and used this model to analyze data coming from patients undergoing antiviral therapy. And you can see the number of viruses falls in this really interesting two-phase manner. And without going into any details, essentially by connecting these two timescales of the decay to model parameters, they were able to develop, rationally develop regimens of therapies using combinations of antiviral drugs that gives you the best shot at ultimately clearing this virus. And they also learned a lot about HIV biology in the process. So again, so this is, I think about it, 5 PCRs and an ODE essentially saved and extended thousands of lives. So now fast forward to today, where now our ability to measure what's going on in a person's body during this infection has just completely exploded, right? We now have the potential to, measure every gene in every cell of the immune system, right? And we can do this over time as well. Here's some single-cell RNA-seq data in HIV patients over time. And these kind of data have revealed a lot of really interesting processes and important processes going on in the immune system during this infection. But from the perspective of quantitative theory, right, I think it's still very much unclear exactly what we do with this data, right? How do you take data about a single cell and put it into a model of an infected person, right? How do we do the equivalent of what Perelson did with 5 PCR data points and his ODE? How do we do that with this flux of single cell data coming in? So I don't have answers. I'm not going to give answers today, but these are the kind of questions I'm thinking about, not in the context of HIV specifically, but more broadly, this question of immune responses and infectious diseases. So just some challenges that have stood out to me is that I think we still don't understand well spatial structure, cellular heterogeneity, and single-cell dynamics. These are all things that we have frameworks for thinking about in general, especially coming from in vitro experiments, but we still don't really understand what's going on in vivo during a lot of important situations. And so what do we do about this? My personal opinion, I think we need to develop more, go back to model systems and really develop more tractable in vivo infection models and just dive in and tune knobs and test predictions like we're used to doing in physics, and in general learn how to kind of coarse grain from single cell behaviors up to the macroscopic level. So that's what got me interested in fruit flies. And so ripping off of Stefan from earlier here, this is my hydrogen atom of immunology, which is basically looking at systemic bacterial infections in fruit flies as a model of sepsis. In fact, I study fruit fly larvae because they are optically transparent. So this gives us the ability to do live imaging and just go in and watch immune responses in real time. And we can leverage the powerful genetic tools available to Drosophila to do precise perturbations and test our theories. And in general, Drosophila has a really well-characterized and highly conserved innate immune system that is strikingly similar to humans. So I'll tell you a little bit about that. The fly has 2 main strategies for controlling bacteria, a system of antimicrobial peptides and a system of phagocytic cells. So antimicrobial peptides are small molecules that are secreted out and damage bacteria, often by poking holes in their membranes. And then phagocytosis, these are cells that are very similar to mammalian macrophages that crawl around and engulf bacteria. During my postdoc so far, I've developed imaging methods for visualizing these cell populations at a really large scale using light sheet fluorescence microscopy. So here, are showing these two populations. At the top, you have the fat body, which secretes antimicrobial peptides. It's this long sheet-like structure that extends along the length of the animal. And then in the bottom image shows the hemocytes, which live in these clusters that are organized into bands along the length of the animal. So essentially, with this kind of imaging, what we're looking at is essentially every cell in the immune system of this whole animal that we can follow. And so just for the rest of the talk, I just want to give you a kind of high-level overview of the kinds of things that we're trying to do with this system. It's still early days. But one of the first things that we're trying to understand is this phenomenon of spatial patterns of antimicrobial peptide expression. So one of the first things I did with this system was to look at a GFP reporter of probably the most well-characterized antimicrobial peptide called diptericin. So what I would do is take one of these larvae, inject them with a non-pathogenic E. coli strain just to activate the pathway, and then just came back 24 hours later to look at the expression patterns. And what we see is that initially the bacteria very quickly are mixed everywhere throughout the animal due to the beating heart, but we see a non-uniform pattern of expression with a bias towards the anterior on the left, shown in green. And we can quantify these patterns. We do cell segmentation based on the membrane markers and add up the intensities and see a nice drop. So one of the things we're doing is trying to understand this pattern of expression. We have a lead on a hormone that is secreted from an anterior region right near where we see all this expression that is a known activator of this pathway. And this is work in progress. The other thing that we're trying to do is from the quantification of these kinds of images is to take seriously these ideas of cellular variability and say, okay, let's ignore space and just plot essentially a histogram or the probability distribution of expression levels across all cells, right? And just try to see what kind of structure are in these distributions. And we find very kind of broad, essentially single peak distributions. And what we want to do is essentially understand why these distributions have the shape that they do by connecting it to single cell dynamics. So kind of very similar to a lot of other contexts, but what comes to mind is, for example, transcriptional bursting, where you can essentially infer parameters that are happening inside single cells during transcription based on the shape of the distribution. We're trying to do this for this kind of in vivo immune response setting, where maybe there's some signatures in the kind of single time point expression distribution that can tell us about what single cells are actually doing over time. And in some very preliminary data we see from cell tracking, we see that cells can have different rates of production. And the last thing I'll tell you about is moving beyond just kind of simple non-pathogenic E. coli to a really interesting bug called Providencia rettgeri, which is a natural fly pathogen that was isolated from the wild and then engineered with nice fluorescent tags by my collaborator Brian Lazaro at Cornell. And so here I'm showing you a just animated Z-stack just of a single time point showing these bacteria. So the only thing labeled here are bacteria. Maybe you can see single cells kind of flickering because they are quite motile in vivo. What's really interesting is that these cells end up in the bacteria end up inside of fly cells. So I'll pause it right here. You can see these groups of small roundish cells down here. These are the hemocytes, or the macrophage-like cells. So these cells phagocytose bacteria. But there's also these larger, more angular cells right here that the bacteria end up into. And we don't know what these cells are, but Brian thinks that this could be an interesting kind of initial point of spread of the infection. So other interesting things about this bacteria and why we want to study them is that, for one, they cause a sort of intermediate level of pathogenicity in that some flies survive this infection and control the bacterial population, and other ones die. And it's not entirely clear what drives this, but it has to do with something about the initial activation of the immune system. And more about that, the fly has about 20 antimicrobial peptides that it used to combat systemic infection, but rettgeri seems to be resistant to all of them except one, this one diptericin that I showed you earlier. So it offers this really unique opportunity to study a minimal host pathogen interaction and kind of manipulate it from both sides. And so what I'm hoping is that by kind of using this live imaging approach, we can just study each part of what you might construct as a kind of simple mathematical model of all of both the infection and the immune response process. and then use our genetic perturbations to test it. And overall goals, again, we want to learn, see if we can infer single-cell dynamics from static snapshots of the infection distribution. And then also, if we learn how the immune system feeds back onto the bacteria, look for dynamical signatures in single-cell data that predict future outcomes. So with that, I want to thank advisor Anand for coming on this wild immune response ride with me, and great students Hennessy and Tiffany who have helped me with this. And I'll leave you with just when you start taking movies of inside of animals, you just start, observing strange things. So I'll leave you with a strange movie of some hemocyte group motility that I've been scratching my head over. Don't understand. You see a kind of a group of 4 cells that are existing. One cell joins, and then they all start crawling off as a snake. So we don't really know what's going on here. So I'll take any questions. Thank you.